Hi, my name is AHMAD AL-AMIN MATERANG with Cometics E22B045. I am from Sabah and I am 23 years old. So today I will discuss about the e-portfolio which is subject from the EFT2143, research methodology and scientific writing. So this is my research title, Assessing Diversity and Distribution of Aquatic Macroinvertebrates in Latter Garden jelly. So this is my objective, to assess the diversity of aquatic macroinvertebrates in Latter Garden jelly and to evaluate the distribution of aquatic macroinvertebrates in Latter Garden jelly. For the following statement, the variety and distribution of aquatic macroinvertebrates in Latter Garden jelly must be assessed in order to understand the ecological health and biodiversity of the freshwater environment. Despite their ecological significance, there is no detailed information on the species composition, abundance, and geographic distribution of this creature in the Latter jelly. This study will be address this gap by truly examining the variety distribution pattern of macroinvertebrates in the region, finding factors impacting their number, and assessing the overall health of the aquatic ecosystem. For the significance of study, the significance of the study lies in its potential to fill up critical gap in the scientific understanding of aquatic macroinvertebrates in Latter Garden jelly, an area that has not yet been researched. By collecting and analyzing data on the diversity and distribution of this organism. The study will provide valuable insight into the ecological health and biodiversity of the region aquatic ecosystem. This information is essential for informing conservation efforts, developing sustainable management practices, and understanding the impact of environmental changes on the local biodiversity. Furthermore, the finding could serve as a baseline for the future research and monitoring, contributing to a broader understanding of freshwater ecosystem in similar unstudied regions. Next is about the literature review. The first one is macroinvertebrate. Macroinvertebrate visible to the naked eyes, lack backbone, and can be aquatic or terrestrial. Aquatic macroinvertebrate living partly or fully underwater include flatworm, crawfish, snail, mollusk, and insects like dragonfly. Second is community structure of aquatic macroinvertebrate. The structure of aquatic macroinvertebrate indicates water body health and is influenced by abiotic and biotic factors. Species diversity and abundance determine community structure, which requires intensive identification efforts. Third is macroinvertebrate composition as an indicator of river health. Macroinvertebrate composition reflects river health with sensitive species indicating high water quality. Pollution sources include wastewater and sediment. Habitat characteristics also affect their presence. What is the role of aquatic macroinvertebrate in water quality assessment? Aquatic macroinvertebrates are crucial for water quality assessment due to their specific habitat preferences and pollution tolerance. Their presence or absence indicate water quality and ecological health. Is functional and structural integrity of aquatic ecosystem. Macroinvertebrates maintain river integrity by decomposing organic matter and recycling nutrients. Environmental factors like oxygen level and pollutant impact their community structure and functions. Next is role of aquatic macroinvertebrate in integrated pest management. Aquatic macroinvertebrate as natural pest predator control pests in agriculture field, farm management practice, and agrochemical use affect their community diversity and pest control efficiency. Lastly, evaluating environmental fluctuation and pollution in aquatic ecosystem. Macroinvertebrate ideal for specific biomonitoring reflect environmental changes and pollution level through their community composition and bioaccumulated matter, providing insight into ecosystem health. Active biomonitoring involved using a cache organisms to assess pollution exposure. Next is methodology. The source of image used in this study is from the Google Earth. For study area, this research is conducted at Latter Garden in Delhi, Kelantan. This location is chosen as a research area due to its popularity as an ecotourism destination in Kelantan. Latter Garden is located at coordinate north 5 feet 44 feet 48.1 inch to north 5 feet 44 feet 48.4 inch and east 101 feet 50 feet 53.7 inch to east 101 feet 50 feet 53.6 inch. Targeting is selected from this research area because it is a new development area for directional purpose. For the data analysis, the Med-Whitney Channel Diversity Index and Simpson Diversity Index are used to analyzing macroinvertebrates in the data getting jelly. So the picture below is the formula for this data analysis. For sample collection, the sample collection used in this work is taken from the Card Aquatic Insect Stream Sample Protocol. Each station has 3 sample points, the right bank, the middle, and the left bank. All 3 samples from each sampling station are combined into one sample, and each station records coordinates using a GPS tool. Samples are collected using a subnet. The subnet is used to collect quantitative data on aquatic macroinvertebrates. For the sample preparation, move the preserved specimen from the collection container to clean and label its container. Rinse the sample with the water to remove the ethanol. For the species identification, sample has been determined to the genus label with the biological binocular microscope model Y100 regulations. In laboratory analysis, all collected aquatic macroinvertebrate specimens are preserved in 80% ethanol. In the laboratory, the preserved samples are examined and aquatic macroinvertebrates are identified to the species level using a standard taxonomy key. The subnet which is used during sample collection allows for quantitative data on aquatic macroinvertebrates. This data is used to estimate the density and abundance of different species at each location has been recorded using GPS tool. The preserved samples are sorted in the lab, and individual macroinvertebrates are quoted and classified into their respective taxa. The Med-Whitney Channel Diversity Index and Simpson Diversity Index test has been used to assess the different macroinvertebrate composition across different locations. The indicators calculated are interpreted to determine the ecological status and water quality of the river, reflecting the preserved and abundance of population-sensitive and pollution-tolerant species. And lastly, the monthly data is compared across different sampling sites to assess temporal changes and species patterns in macroinvertebrates community. So this is my milestone, which is start with the research proposal development, 17 March until 25 March. Research equipment procurement, 26 March until 4 April. Site visit and data collection, 5 April until 20 May. Laboratory analysis, 21 May until 31 May. Data interpretation, 1 June until 10 June. Final report and documentation, 11 June until 20 June. Review and presentation of finding, 21 June until 25 June. And lastly, project evaluation and closure, 26 June until 30 June. So this is my Gantt chart is explain about my progress in the e-portfolio, which is started at the Create a Google site at the week 4 March until final submission at the week 4 June. So this is my research budget, which is started with fieldworks, transportations, food and supplies, equipment, subordinate software train, field notebook, GPS device, laboratory analysis, microscope, sample preservation material, and chemical reagent, data analysis, statistical software license, and lastly, publication, printing, and binding report. The total of this budget is 8,258 ringgit and 81 cents. And lastly, references. The source of the reference is from the Research Guide and Science Directs.